Hi, first of all, thank you for showing an interest in my work. My name is Gerald Renten and I will talk about my work which is titled Altered Response of the Functional Network during a Pain Stimulus in Small Fiber Neuropathy. I have nothing to disclose. Small fiber neuropathy, or SFN, is a neuropathic disorder associated with chronic pain. And around 5% of these patients harbor a mutation in the SCN9A gene, which we now refer to as genetic SFN. Chronic pain impacts the brain, and it may especially affect the functional organization of the brain. Therefore, in this research, we will investigate what the potential effect of SFN-related chronic pain is on the functional organization of the brain network. And moreover, we will assess whether this effect is different for the genetic variant of SFN. Our sample comprises of 20 idiopathic SFN patients, 11 SFN patients with the genetic variant, and 20 healthy controls. For all the SFN patients, we obtained a measure of self-reported current pain using a visual analog score. All participants underwent 3T MR imaging, and we obtained T1 weighted images and 2 fMRI scans. One fMRI scan was with a heat stimulus, so we applied 7 heat stimuli of 12 seconds alternated with 36 seconds of rest. And the second MRI scan was similar to that, but the stimuli was not painful, but it was a non-painful stimuli. So we could also assess the effect of stimuli which is not related to pain. For the pre-processing, we first parcellated the T1 weighted image into several cortical and subcortical regions using FreeServer. These regions could subsequently serve as our nodes in the network, in the functional network. The functional images were corrected for slice timing, and they were corrected for subject motion using realignment, and they were bandpass filtered, all using FSL. Next, the first 5 dynamics were discarded to assure steady state of the signal, and the time signals of each region of interest, which was previously defined using FreeServer, was corrected for the head motion parameters and the signal of the white matter and the CSF using regression. Finally, the functional network was quantified using the Pearson's correlation coefficient between all the previously mentioned regions of interest. Since network analysis is very dependent on the number of edges in a network, we made sure that we only compared networks that were equally sparse. And the graph measures that we calculated are the path length and the clustering, which are 2 of the most robust and most commonly used graph measures. And they are measures of the global network, so they quantify the whole network in one go. And then we also looked at a local graph metric, so which is on a nodal basis, and for this we used the betweenness centrality, which is a measure of nodal importance of the node. So it's calculated as how many times a certain node is included in the shortest path in that network. So what we found was that the cluster coefficient was higher in patients with small fiber neuropathy compared to controls for the fMRI scan with the heat stimulus. And for the path length, we observed that the genetic variant was higher compared to the healthy controls and not the idiopathic SFN. Moreover, when we look at the non-painful stimuli, we see that the patterns are similar, although we don't find any significant differences in this non-painful stimuli. Moreover, further investigating the local measure, so the betweenness centrality, we found that the betweenness centrality of the postcentral gyrus and the thalamus are significantly lower during the painful stimuli compared to the cold stimuli, and we only found this in the SFN group, so the people with chronic pain. It's not observed in the healthy control group. When we further investigate this, we found that during the heat stimulus, the betweenness centrality, so the nodal importance of the post-central gyrus, relates differently to the self-reported pain score compared to the cold stimulus. So since the non-painful cold stimulus did not show any significant differences between the small fiber neuropathy patients and the healthy controls, this indicates that our results are not purely driven by sensory input, but are more specific towards pain and the pain pathway in the functional brain. A previous recent study also showed that functional brain network exhibits less segregation and more integration during a painful stimuli. And combined with our result, this indicates that the cerebral network plays an essential role in the perception of pain. And that relates then, or that is in terms of a increased segregation and decreased integration in patients with chronic pain. Thank you very much for your attention. If you have any questions regarding this work,